The stories that you're telling. And her lectures are like demonic devices. I love how she like explains that and like says that like form that. Maybe she said they're both doctors. Yeah. That's really cool. No, I know. How funny would that be? They're just like,
Okay, for whatever reason, I didn't like the way it was set, but looks like it's okay now, so here we go. Maybe. Slowly, but yeah. Your favorite diagram, right? Great memories here. So it was, you guys found an example, and then we measured values. So if we look at each one of these, we start with chylomicrons. Remember, chylomicrons are made in enterocytes. That's where the dietary lipid is incorporated before it goes into the lymph and then the blood. And as it travels through the blood, it interacts with lipoprotein lipase. And that lipase hydrolyzes triglycerides, turns them into fatty acids and glycerol, which absorb into memory tissue, muscle tissue, adipose tissue. And of these, most of that dietary lipid ends up in adipose tissue. In order for it to do that, chylomicron needs ApoB for structure. Remember the other A protein? C2. ApoC2, that's one that acts activates the lines. So you got D48, D2. I'll tell you why that's important. And chylomicron popped off most of the triglycerides, you cycle through all. And then chylomicron goes to the liver. From the time chylomicron into the blood, and it's metabolized, they take a few hours. So that process occurs about 4 to 5 hours, going from enterocyte into the lymph into the blood, and then it's gone. So you guys fasted for 12 to 14 hours. Chylomicrons are gone. So you're not measuring that in your serum. So I measure the lipids we see. Okay? We're more interested in that protein stay around for longer, which means so the elements. Now, in these lipids, remain in the liver are incorporated in the LDL. The LDL have mostly triglycerides, some cholesterol. They go directly into the blood. Remember the 8 proteins they have? ApoB 100 and C2, yeah, good. You need C2 to interact with lipoprotein lipase also. They drop off their triglyceride, they retain the cholesterol. They're then called VLDL remnants. Those remnants are either taken up by the liver or become LDL. LDL are mostly cholesterol. And the main function of LDL is to deliver cholesterol to other tissues. That's what this is showing, the extrahepatic tissues. Now some of that LDL becomes incorporated into atherosclerosis, those plaques that we talked about before. So that's one of the downsides of this. One of the reasons that tends to happen is when those LDL are modified, they become the targets of those macrophages that are in the arterial intima around the cardiovascular system, especially. LDL stay in the blood for 2 to 3 days. They stay in the blood, tend to stay in the blood longer than anything else. So their likelihood of being modified is affected by it. The longer they're in the blood, more likely they are to be modified. The VLDL clear pretty quickly as well. So they go from the liver to capillaries and tissue within a few hours as well. So when you measure your serum lipids like you guys did, very little of that is VLDL. Most of it's LDL and HDL. HDL, they're mates, remember down here, they start the liver and the intestine, but they come out as little small molecules that are relatively flat. They come out as like little discs. As they accumulate cholesterol, they become spherical and larger. So they pick up cholesterol in extrahepatic tissues, deliver it to the liver, most likely for excretion. Okay, so that's why we say high HDL is good, high LDL is bad in general. Okay, now here's an important question. The number one lethal disease in the U.S. is heart disease, cardiovascular disease. Why are humans so susceptible to heart disease? One major factor has to do with ApoB. In humans, chylomicrons contain ApoB48, and LDL contain ApoB 100. Now, the form of ApoB significantly impacts lipoprotein metabolism. Lipoproteins with ApoB48 tend to be cleared faster than those with B100. Both forms of ApoB are made from the same gene. ApoB48 and ApoB100 come from one gene. Two different proteins, one gene. So how are both ApoB forms made from one gene, and how does it relate to heart disease in humans? That's what we're going to talk about on this process called ApoB mRNA editing. I don't know why that slide is off. It says ApoB mRNA editing occurs in human small intestine. It should say small intestine. That's weird. Here's the ApoB gene. Right here. So this is DNA, 5', 3'. Here's a central codon in that gene. It has a sequence of CAA. Down here, at the 3' end, we have TAA. What's TAA represent? Stop codon, remember that? CAA encodes for an amino acid. When transcription occurs, remember, an RNA molecule is made and you end up with an mRNA molecule. In this case, we have what's called no editing. That means the transcript sequence is from the gene. The central codon remains CAA. Here's our stop codon. Thymidine is converted to uridine. So now we have a stop codon here. So now when translation occurs, translation starts here, 5' end, goes through, all the way through the sequence until the stop codon. So you end up with a full-length protein. That protein is referred to as ApoB 100. That means 100% of the transcript is translated. That's different than this scenario. And again, I think I'm missing some text on this. Shows CAA converted to UAA. In other words, the codon representing an amino acid is edited to a codon that represents a stop codon. So what happens? Translation starts, goes about halfway down the transcript, and stops. That means you end up with a protein that's about half the size. More specifically, 48% the size. That's why it's called ApoB 48. Simple? That happens in the nucleus. So if editing occurs, it happens in the nucleus.
Oh, small intestines.
So it'll happen in the nucleus of the cell, so basically it's post-transcription, pre-translation, right? So the transcription occurs, and then the RNA is sometimes edited. Here's a little bit more detail and a slightly different way of looking at it. This is ApoB mRNA editing. That's the process. ApoB mRNA editing involves the CDU conversion performed by a cytidine deaminase, and that is an enzyme known as ApoB1. So there's a category of enzymes that are called cytidine deaminases, and you can imagine what they do. They deaminate cytidine. So if you remove an amine group from cytidine, guess what you get? Uridine. It's a CDU conversion. The enzyme is known as ApoB1, and that stands for ApoB mRNA editing catalytic subunit 1. Simple, right? I think the reason they called it ApoB1 is they thought there were going to be more of these, but they only found one. ApoB mRNA editing catalytic subunit 1. Now, again, another way of looking at it, here's codon 1, here's codon 4564, central codon right here, codon 2153 in the gene. This is in humans. In the liver, the transcript is unedited. So translation is normal. You end up with a massive protein, 4,563 amino acids. Function transport of cholesterol in the blood. In the intestinal cells, the mRNA, the unedited, is converted to an edited. So CAA, the glutamine, is not encoded for, change your stop codon. So during translation, you end up with ApoB 48. Function, absorption of lipids from the intestine. That's the way humans do it. Why do we edit in one tissue but not the other? Tissue-specific expression of ApoB1. Bottom bullet point. Tissue-specific expression of ApoB1 determines whether or not editing will occur. So do intestinal cells express ApoB1?
Yes.
Yep. Do liver cells? Nope. Now, this has a huge impact on our lipoprotein metabolism. Because if you think about it, if we express ApoB1 in the liver, we can make VLDL with ApoB 48. And I said that ApoB48 containing lipoproteins are cleared faster. If they were cleared faster, less chances of modification, that could significantly decrease the amount of cholesterol in both our LDL and our blood. Let me give you the background of this. Once they figured out that ApoB1 was the enzyme that did this, and I remember these papers because when I was in grad school we were doing research on this. There was a lab at Baylor and another lab that I think was, who else cloned that? I think they were in New York. Same time they came out with this paper that talked about ApoB mRNA editing. It was the first time that they had ever characterized something like this in mammals. That made it a big deal. The other thing was, as soon as they did that, they realized, you know what? If we could express ApoB1 in humans utilizing gene therapy, we could potentially significantly decrease cardiovascular disease. So these massive labs, multi-million-dollar research labs, started going after this. Their goal was to clone this enzyme, figure out a way to induce its expression in people so that we could significantly change lipoprotein metabolism and decrease cardiovascular disease.
Would it affect like actual body composition or just like the cholesterol levels?
I don't know about body composition.
Okay. Like there's people would solve like other problems, but it's like that was cholesterol.
Yeah. Oh, I see what you're saying, yeah. But potentially you could say, well, even though they are, let's say, they have other risk factors like obesity, we can still decrease their cholesterol, their LDL cholesterol. Okay? So here's how, this is what you have to do. If you're going to figure out how to make this work, you got to understand how it actually works in the body, because then you can decide what you need to do in order to induce this function in human liver. Have you guys ever heard of this, like gene therapy? You know what that is? If someone, for example, has an altered nucleotide sequence in a gene, like a classic example is PKU. So when I was doing research at Oregon Health and Sciences University before I came here, we had our research lab, and right next to us, one of their research projects was PKU. So they were looking at gene therapy and ways to induce a normal gene in the livers of people that have PKU. So they took the phenylalanine hydroxylase gene, incorporated it into a DNA vector, which is basically a piece of DNA. Typically, the vectors that you use for these types of things are viruses. One of the most common vectors used to be adenovirus. It's a DNA virus where you can incorporate your gene that you wanted expressed, and then you infect people with it. What you do is disarm the virus. You take out all the harmful components of the virus, but you use it to be able to go into tissues, because once it goes into cells, it can integrate into a person's DNA. You guys have all learned a little bit about that, retroviruses and all that. Well, adenovirus is a double-stranded DNA virus that can do the same thing. So they can put genes in those, infect people with it, and express the gene that's in there. Now, they haven't figured out PKU exactly yet, but that lab was actually had some pretty interesting data on mice. And I don't know whatever came out of it. That's been years ago, but that's the type of things that they were going to try with this.
I feel like, wouldn't that be so dangerous? Because there's got to be a reason why this enzyme is expressed in our small intestine and not in the liver. Have they researched like possible things that could go
Oh
wrong?
yeah, so this is what happened. And we actually were collaborating, at this point we started collaborating with the lab in Baylor. So we knew some of the inside stories on what was going on. And they had researchers from all over the world. The woman that cloned this protein was from Taiwan. Her name was Bobby Tang, and she was extremely sharp. She had cloned this, and then another guy in their lab named Paul Lau figured out that it was actually a dimer, that the enzyme binds to itself, another copy of it, and then edits. I don't know how much they were involved in these studies, but they tried a couple clinical studies where they took this, incorporated it into a virus, and infected people. They did this after doing it in animals. In animals, and I believe in 2 people, they quickly found out there were problems. Here's what happens. This ApoB1 is a dimer. remember Paul wouldn't tell us the title of his paper till it came out. It's like, I'm not going to tell you. Just tell us the title. I can't tell you. Tell us the first word of the title. I can't tell you. Whatever, Paul. This was it was something like homodimerization of ApoB1 is required for its function. ApoB1 forms a homodimer. Okay? ApoB1 requires a complementary factor. That's this. See this big blue area here? That's another protein. So ApoB1 is a homodimer. What part of the cell are we talking about? In the nucleus. So ApoB1 goes into the nucleus, forms a homodimer, and interacts with this protein. See where it says P66? Two labs named this at the same time. One lab called it P66 because that's the size of it. It's a protein that's 66 kilodaltons. Another lab called it auxiliary complementary factor 64. Whatever. We call it complementary factor. This protein provides sequence specificity to ApoB1. So look at this protein's doing. It found this sequence. It binds to this specific sequence. It binds to ApoB1. So basically, the complementary factor finds the appropriate sequence for ApoB1 to edit.
Sequence specificity?
Yeah. The complementary factor provides sequence specificity for ApoB1. So this is the cytidine that needs to be edited. But as you can imagine, there's plenty of cytidines in mRNA, because if you look at this sequence, there's another one right here. Why not edit this one? So ApoB1 has to do the right cytidine. When they did those studies with the adenovirus infection, editing lost its sequence specificity. In other words, ApoB1 was editing other transcripts. It increased the risk of cancer. So in mice, they had increased tumorigenesis. I don't know about the people. I know they stopped those studies immediately. It was a big deal because at the time, we were all chasing this. What we were doing is we actually cloned this in hamsters. So I had a paper a few years ago, Journal of Nutrition, where we cloned this ApoB1 in hamsters and compared its sequence to some of the other ones. And what's interesting about hamsters is hamsters have a similar lipoprotein profile as us. They have high LDL cholesterol, low HDL cholesterol. Rats are the opposite. Rats are HDL animals. They have more cholesterol in their HDL than their LDL. Rats don't get heart disease. They don't get atherosclerosis. So a huge factor in whether or not a species gets atherosclerosis is whether or not they make ApoB1 in the liver. So everybody's chasing this, and all of a sudden we went, oops, dead end. We didn't care because we were working on other parts of this. We were looking at how things actually regulate this. But those big labs, basically, the whole bottom dropped out of this area of research. And they dropped it because they went, wow, no heart disease but cancer. We call that productive. So they basically stopped. Now, if this ApoB1 is expressed, that tissue edits. So we know that in people, we express ApoB1 in intestinal cells, right? We don't express it in the liver. What about rats? Do rats express it in intestinal cells?
Yes.
That's what they put in their chylomicrons. Do rats express ApoB1 in the liver? Yep. They do, we don't. So we used to do assays where we would take rat liver and you can actually quantify the percentage of ApoB mRNA that's edited. And rats in the liver edited approximately 50 to 60%. In other words, about half of the VLDL in rats have ApoB48. About half of them have ApoB 100. The ones with B48 are cleared quickly. They don't become LDL. The ones with ApoB 100 can become LDL. Now what about what I just said? How about hamsters? Do hamsters have hepatic editing?
No.
They don't. And when we would characterize their lipoprotein profile, it was pretty similar to ours. Which is kind of unfortunate for them. One of the things we did was we also sequenced this right here. And we looked at the sequence, it says Maureen sequence. This sequence is really important for the complementary factor to find it. So when we line this up and compared it to rats and other species, really similar here. But other parts of the transcript could be slightly different, doesn't matter, but this almost has to be perfect. That's how the complex finds the appropriate sequence. So that's a big effect on our lipoprotein metabolism. That's why we have high LDL cholesterol values. This is all review information, right? We've seen that before. So we know that chylomicrons, VLDL, triglyceride rich. LDL and HDL are cholesterol rich. Now, we looked at this before, but now let's kind of put your values in the context of this table. This is distribution of triglycerides and cholesterol among fasting plasma lipoproteins. So here's total. It says plasma, that's total cholesterol. And remember, you guys did serum, but it's very similar. You have total cholesterol approximately 176. This is mg per deciliter, same units as you're using. VLDL, very little, about 5% of the cholesterol is in VLDL. Why? It's cleared, it's converted. This is fasting. It's had time to be metabolized. IDL, even less. Look at LDL, about 115 of the 176. That represents 65%. Humans are LDL animals. We have most of our blood cholesterol, typically, in LDL, the particle that can hurt us the most. HDL, kind of a typical value, about 45. About 25%. Now what I want you guys to do today is you're going to do your triglycerides. You're going to use that to calculate your LDL. So at the end of the day, you'll have your total, your HDL, and your LDL. Your LDL plus HDL should be approximately what percentage of total? 90. So add yours up. That's kind of a way that we can check each of your assays. Your LDL plus HDL should be approximately 90% of your total. Okay? Now, some of you guys are going to have similar values to these. Some of you will have higher LDL. Some of you will have lower HDL. Some of you are going to have similar values. Your LDL is going to be similar to your HDL. Good for you. That can be very protective. Okay? Some people actually have higher HDL than LDL. Kind of rare, but it happens. It wouldn't surprise me if one of you were in that case. I've even seen it like in our research lab. I looked at all the lipids values for the people in that, and some of those women had a higher HDL than LDL.
Is that genetics more? Is that
I think in general for this, we have kind of a genetic window for lipids, and we can do things to kind of modify our levels within that window. But overall, yeah, some of that's genetics because there's lots of things involved here. Your ApoB 100 sequence, your lipoprotein lipase sequence, the receptor sequences. So it's, yeah, that's a big part of it. Over here, triglycerides. You guys are going to do triglycerides. totals. Average might be something like 85. Even though most of the VLDL is cleared, remember VLDL is mostly triglycerides, so if any of it's there, that's where it's going to be, the triglycerides mostly there. So when you see your triglyceride values, remember most of that's in VLDL, about half of it. Why are we doing this? We're doing this because you can take, so what research labs did years ago is they separated these using ultracentrifugation, each of the lipoprotein subclasses. They were able to determine a factor. If you take your triglyceride and multiply it by a certain factor, put it in an equation, that gives you your LDL cholesterol. Again, this is the way clinical labs do it, the way you guys are doing it today. Research labs, you tend to do it a little bit differently, but they're both accurate. So that's why you guys are doing triglycerides. Otherwise, we would have to spin down your LDL, and that's not easy. I mean, that's an overnight spin, usually, at about 70,000 RPM in a special centrifuge. So we won't do that. The other thing is triglycerides are also a risk factor for cardiovascular disease. Okay? One thing to keep in mind today, when you're doing your assays, there's going to be high variability. Some of you might have 25, some of you might have 150. Make sure you put your cuvette in the spec correctly. Jamie, while I'm thinking about it, could you turn these on? Okay, so you guys are going to do that today. Now, remember these tables? Blood cholesterol levels, what do we want? Well, here's desirable. We want low total cholesterol, low LDL cholesterol, high HDL. Borderline elevated risk, if it's between 200 and 239 total. For LDL, if it's 130 to 159, HDL, 40 to 60. Elevated risk, greater than 240. Greater than 160 LDL, less than 40 HDL cholesterol. And these are highly variable. Like I remember a few years ago, a friend of mine came to me, and he's about 60, early 60s. I don't think he'd ever had his blood work done. He showed me his blood work, and it was actually, his numbers were bad. His HDL cholesterol was like 25 or 26. That's a low value. And he was asking me about it. And I said, yeah, that's not a good value. I'd have it retested just to make sure. But his diet was terrible. His exercise was not good. Had extra body weight. And I think genetically he was kind of in that case. Now, you guys know your blood lipids at a nice young age. I would encourage you to get your parents, encourage other people that you know, have their blood work done so that they can find out those values. It's nice to know, and sometimes if you need to make changes, you can. A person doesn't want to have altered blood lipids for years and not know it, like that guy I was talking about. That's the first time he probably ever had it done. So it's better if we know early on. Now, again, multifactorial. So just because a person has high blood cholesterol doesn't mean they're going to get heart disease. There's lots of other factors. For example, if they have a previous heart attack or diabetes, that tends to elevate risk. This is for the risk of cardiovascular disease, or two or more of the following. Very high risk would be smoking, high blood pressure, for example. Smoking is almost the worst. There's not many things you could do that's worse for your health. High blood pressure, low HDL, family history of heart disease, early heart disease, and age. For men, it's over 45. For women, it's greater than 55. Remember, one of the scenarios here is HDL is protective. Women tend to have more HDL, and one of the reasons for that is estrogen tends to increase HDL production. So post-menopause, for a lot of women, their lipoprotein profile changes dramatically. In general, their triglycerides will go up, their total cholesterol will go up, their LDL cholesterol will go up. For men, we're more about chronically suffering, right? So ours doesn't change much as we get older. It tends to stay the same. In general, though, as we get older, blood lipids can go up. So it's good to know those So what are typical values? That's kind of what we talk about, those values, but is that what people actually have? So they've done obviously lots of large studies, so I pulled one of those out that the results were published a few years ago. This is a Minneapolis-St. Paul study. They followed 2,500 subjects for about 20 years, and some of their findings were kind of interesting. So again, they did this a few years ago, and let's see, they had men and women in the study. The first readings that they took were 8 at 82. You guys weren't alive yet. It was too bad. The 80s were awesome. Okay. 80, 82. For men, they had total cholesterol of 212. 80s were fun, all right? And then,
Let's hear some stories.
208. Probably shouldn't. 20 years later, same group, same cohort. Took them again. A lot of stuff going on these days, too. Part of it was, education programs, lifestyle, diet, things like that. So what do you think? Did their cholesterol go up or down? It's going men. Women. Up or down? More than the men?
No. Yeah.
Yeah, I figured you'd say that. 197. Come on, that's pretty similar, right? Why'd you go down? Yeah. Any ideas? Good lifestyle, good. We're listening to everything we're being told, taking care of our health. Could be, so good point, but we're assuming, maybe same assay, same variability.
It's the same cohort.
Same cohort, yeah.
They might have been actively working on it.
Could, yeah, absolutely they could have been. Yep. You guys ever watch? You guys don't watch what I watch, I'm sure. If you watch some of the things I watch, they're targeted at a certain demographic. You see a lot of commercials. I've mentioned this before. New car commercials, typically Buicks or Jeeps. You see investment companies, and you see a lot of these. Crestor, Lipitor, lipid-lowering medications. Is that common? Extremely. Incredible amount of money in these. Their hypothesis is this went down because of increased prevalence of lipid-lowering meds. And the reason they said that is they could see increased incidence in certain age cohorts, but not in all of them. Individuals 25 to 34 years of age, when they track them over time, their values didn't go down. Matter of fact, they might have went up a little bit. Why? They're not on the meds. And a lot of them aren't because they don't know what most people don't know what their lipids are early on. A lot of times when you're turned 50, I gotta start taking care of myself. Right? Turn 60, people start being concerned about that. So their values in an age group just slightly higher than you guys actually kind of are still going in the wrong direction. Okay? So lipid-lowering medications, we'll talk about that. So that's why we think that total cholesterol might have dropped slowly because of that increased use of medications. Exceptions would be younger groups that aren't using those medications. So the conclusions are blood cholesterol values are important to consider. However, they should be considered with other aspects of health, like blood pressure, smoking, exercise. Another issue is blood glucose, right? Those can kind of go together. That can have an impact on risk of cardiovascular disease. Even if blood lipids are normal, they should probably be repeated tests every few years. If they're elevated, probably more frequently than that. But I'd encourage you, again, get the word out to anybody you know. They should know their blood lipid values. How many of you guys know that your parents get their blood lipid values tested? Okay. This is why it's so important. Remember the response to injury theory. Causes of injury include hyperlipidemia. It's basically what we're talking about today. We didn't do your blood pressure, and I'm not going to ask you to smoke. But we know your blood lipids. Steps in the process. Remember, there's the injury to the endothelial cells, macrophage accumulation, fatty streak formation, because those macrophages are consuming the LDL particles. That can lead to plaque progression and eventually plaque rupture. So one of the most important things is high plasma LDL levels are a major factor in the development of atherosclerosis. So what can be done to keep LDL low? Exercise, one of the best things you can do, and we're nutrition majors, so diet. Fiber, right? Remember that the type of fat that's consumed can affect plasma LDL. This is a little controversial at this point, so I'd say fortunately they're revisiting a lot of this data and doing more studies to look at the effects of saturated fat. But in a lot of people it seems that saturated fat intake affects lipid cholesterol as much or more than dietary cholesterol itself. So it's important that a person keeps both of those in mind. And it can have a significant effect. Unsaturated fat tends to decrease plasma LDL. Dietary fiber tends to decrease plasma LDL. How do those work? So let's look at this real quick. One of the things that causes LDL cholesterol to go down is the amount of LDL cholesterol that tissues are taking up. We said last quarter that once you have LDL, about 75% of them are cleared by the liver. About 25% go to other tissues. With that known, one of the things that companies did was they started figuring out drugs that could actually decrease liver cholesterol synthesis. Because as liver cholesterol concentrations go down, LDL receptors go up. Remember that? As liver cholesterol concentrations go down, LDL receptors go up. So what happens to the LDL particles in the blood? They're more likely to be cleared by the liver. So most of the new drugs target that, liver cholesterol synthesis. How does fiber work? Yeah, remember, so part of it is, if you have fiber in the gut, and it binds to bile acids and or cholesterol, and you decrease the amount of cholesterol that's absorbed, and you decrease the amount of bile acids that are recycled, what does that do to liver cholesterol concentrations? It can go down because the cholesterol is being converted to a bile acid that's going back into the GI. So liver cholesterol concentrations can be driven down by fiber. So what happens? More LDL receptors, more uptake from the blood. So that's a good way to do it, increase fiber. And the other thing, as you mentioned, is production of propionic acid from fiber fermentation. And that decreases HMG-CoA reductase, that decreases cholesterol concentrations in the liver as well. All right, so we want to make sure, and overall in this country, obviously, most people don't exercise enough and don't consume adequate amounts of fiber. So maintaining a healthy body weight and exercise is also important. One of the reasons these labs are back to back is because insulin can have an impact on lipoprotein metabolism. Being overweight or obese can cause a person to have a reduction in insulin sensitivity. So insulin doesn't work as effectively. So what does our pancreas do? Release more insulin. Chronic high levels of insulin can increase hepatic cholesterol production, and that can cause an increase in plasma VLDL and LDL. So insulin's an important part of this as well. Any insulin that we make, we want it to work effectively so we don't have to use very much of it. And I've seen this in research studies where we're monitoring blood lipids and we're also measuring blood insulin. If insulin is up, it's a good indication that person might be pre-diabetic and might be on their way to becoming diabetic. And at that point, sometimes you see an elevation in blood lipids.
Doesn't it stimulate ApoB?
It does, yep. So after binding to its receptor, insulin affects cholesterol synthesis by at least 2 mechanisms. And that's what's here. And the reason we're going through this is this sets us up for the discussion on lipid-lowering meds. So here's acetyl-CoA. And remember, most of this is going to be more relevant in the liver than any other tissue. Here's acetyl-CoA. There's multi-steps here before we make a molecule called beta-hydroxy beta-methylglutaril-CoA, also known as what? HMG-CoA. The enzyme HMG-CoA reductase converts that to mevalonate. Then there's several more steps for cholesterol. That cholesterol concentrations intracellular regulates receptor-mediated endocytosis. So if you have high cholesterol, decreases receptor-mediated endocytosis, decreases LDL receptor synthesis. Let's look at insulin. Insulin upregulates HMG-CoA reductase. Remember, HMG-CoA reductase is the key regulatory point in cholesterol synthesis. Insulin actually increases the expression of the gene, so you get more protein. Not only that, but insulin activates the enzyme by dephosphorylating it. So it makes more enzyme and activates more of it. So what happens? Cholesterol synthesis goes up. What would that do to LDL cholesterol in the blood and why? It would increase LDL cholesterol in the blood because less receptors are going to be on those liver cells because they've increased their cholesterol concentration. Okay, so we want insulin values in general to be low and as effective as possible. Let's take about hopefully about 3 or 4 minutes to talk about these lipid-lowering medications. Now, the work on this class of drugs started in the 70s, and a researcher in Japan that was being funded by a company, you guys might have heard of this company, Merck. Merck is one of the largest pharmaceutical companies in the world, and they started funding this, and this guy figured out that there's a type of molecules called statins. So there's a whole group of these that have similar structure. They have different trade names like Lipitor and Crestor and atorvastatin and quite a few of those. This is how they work. Statins inhibit HMG-CoA reductase by competitive inhibition. They actually bind to the enzyme at its active site and inhibit its activity. Here's an example of one of their structures. I think this is lovastatin. They all look kind of similar. What do you think these guys look like? HMG-CoA. So those drugs kind of mimic HMG-CoA, bind to HMG-CoA reductase, so HMG-CoA doesn't. So what does it do? Cholesterol synthesis goes down. What happens to the LDL in the blood? Goes down. Why? Because the drugs are decreasing cholesterol concentrations in the liver. So the liver cells making less cholesterol, because of that, increases its LDL receptors. So more LDL is taken up by the liver. Sound like it might be effective? I think it can be very effective. Depending on the individual, the type of drug used, they can decrease LDL cholesterol by 50 to 75%. That's purported effectiveness, and so for a lot of people that's the case. Is that significant? That can be really significant. Are these drugs popular? No, probably 40 to 75%. This is a multi-billion-dollar a year business. For the numbers, I think it was like a couple of years ago, Merck said, yeah, we made about $2 billion off of the statins. So it's huge. So it's very, very common that a person goes in and has these recommended to them. As soon as they have a high blood lipid value, a lot of times they'll say, oh, we're going to put you on some statins because we want that LDL cholesterol to go down. Now, ideally, what do we want? Kind of last resort, hey, let's look at your diet. Let's look at your exercise. Let's look at your lifestyle. Do you do all these other things? Let's try to avoid these statins. Why? There's side effects. Some of them can be significant. So, for example, a few years ago, my mom's blood lipids kind of went through the roof. And so we were talking about it and everything, and I don't really know. Again, even your moms only listen when they want to. Didn't really do much about it, and they went up to, 325, something like that total, and they were normally about 200. And fortunately, her doctor said, I don't want to put you on statins, I don't like them. Too many side effects, they can be nasty. So do this and this. And my mom did a lot of changes, and they started dropping. Now they're down below 200. So didn't have to go on them, so thankfully. But a lot of times that's not the case. Let's put you on these. We want that LDL cholesterol down. And one of the interesting things about statins is they are probably more effective in decreasing the risk of a second cardiac event than the first one. So if a person has a history of cardiovascular disease, they're a better candidate for statins than someone who has not. If they haven't had one, and if we can, I think other things want to be done. Some of the side effects that are potentially there. One of the most common side effects is muscle loss and muscle pain. One of the issues with the statins is specificity. So if you think about how are you taking them? Orally. They end up in your blood, they go to tissues. A lot of the statins ends up in the liver, a lot of them end up in other tissues. So one of the theories is that some of those statins incorporated into the muscle cell fiber plasma membranes, the sarcolemma, and compromises their integrity. Different statins do this in different people. So some people will get on one statin, they have problems, they'll switch to another statin that seems to be better. One of the issues is, another side effect is potentially kidney dysfunction. If you're breaking down muscle cells and some of those proteins are leaking into the blood, that can cause kidney problems. If it's bad enough, it can cause kidney failure. It's really important that people on statins are being monitored consistently. The other main side effect is potential liver dysfunction. Same thing, specificity issues where those liver cells are actually damaged. So ideally they're doing liver cell enzyme testing in the blood before and throughout the statin therapy to make sure that's not an issue. Okay, good there? I think we're set. I think I went through everything we needed to. So what we'll do is, let's take about a 10-minute break and we'll run through the experiment real quick. So start gearing up, like get everything ready to do your triglyceride assays. Stop this real quick.
I know, I almost think I'm so sorry. Yeah. I wish I did.